Welcome to class number 3, session number 4. This session will give an introduction for an important sensing approach that is called sensors array or electronic nose. The introduction will give a brief information on the hardware and software parts of this sophisticated sensing approach. So far, the strategies for sensing analytes can be divided to two main techniques: selective sensing on the one hand and cross-reactive sensing on the other hand. In the selective sensing concept, a highly selective receptor or detector is designed to specifically bind or detect the analyte of interest. Please refer to the upper part of the presented schematics for more details on this sensing approach. This approach is suitable for detecting a well-defined target analyte in the presence of interfering species or background. While most selective sensing techniques for detection are aimed for non-volatile compounds, there are a number of well-defined sensors that target specific analytes of well-characterized applications. An emerging strategy that is complementary to the selective sensing approach is the so-called cross-reactive sensors array, as demonstrated in the bottom part of the presented schematics. Bio-inspired, this approach performs detection throughout use of an array of broadly cross-reactive sensors in conjugation with pattern recognition methods. In contrast to the selective sensing approach, each sensor in the cross-reactive array is broadly responsive to a variety of analytes. In the S architecture, each analyte produces a distinct fingerprint from the array of the broadly cross-reactive sensor. This allows to considerably widen variety of compounds to which a given matrix is sensitive, to increase the degree of component identification, and in specific cases, to perform an analysis of individual components in complex multi-component biochemical media. Pattern recognition algorithms can then be used to obtain information on the identity, properties, and concentration of the analyte exposed to the sensors array. In continuation to the concept presented in the previous slide, array of multiple sensors can be used when multi-component gas analysis is required. For instance, if you want to distinguish a good wine from a bad wine based on their aromas only. This approach mimics the olfaction mechanism of human, dogs, and other animals, and it is therefore known as machine olfaction or simply electronic nose. Applications of electronic nose ranges from food industry, medical industry, and environmental monitoring and process control, and many, many more. As an example of the electronic nose concept, I present here in this slide the so-called optoelectronic nose. This device looks a bit like a computer chip covered with neat rows of dots. Each dot contains a chemical dye, and some of these dyes are made of materials that change color to show how acidic or basic a chemical is. If you have ever used the litmus paper, you know how this approach works. This paper contains a dye that turns red for an acid, such as lemon juice, or blue for a base, such as baking soda. In the presented example of the optoelectronic nose on the slide, the resulting color pattern is like a chemical fingerprint. Each pattern is unique to a single odor or mixture of odors. For example, exposing the 5 by 5 sensors array seen on the screen to hexylamine vapor produces colors in different places of the array. Therefore, the produced combination of colors and their constituent places in the array would serve as the signature of the hexylamine. On the other hand, exposing the same sensors array to pyridine showed different combination of colors that is different from the one of the hexylamine. Similarly, other types of analytes, such as those presented on the slide, show different color or optical signature on the same array of sensors, and therefore for each analyte it has its own fingerprint of colors or optical signals. Many types of sensors can be used for electronic applications. Metal oxide sensors are considered as one of the standard sensors. Other sensors are based on surface or bulk acoustic wave, field effect transistors, or conducting polymer sensors. Optical sensor systems is also an interesting option, as seen in the previous slide. Indeed, instead of having transduction principles based on electrical signals, potential, or frequency, the modulation of light properties is measured in this approach. Mass spectrometers are often applied for lab analytics or as standalone devices for the identification of pure chemicals. The disadvantage of all types of mass spectrometers is that their operation requires a vacuum, and therefore they are not as convenient as the solid-state sensors array. With this in mind, the most common chemoresistive nanomaterial-based films are based usually on conductive inorganic nanomaterials, such as metal nanoparticles, single-wall carbon nanotubes, carbon black, that are capped with or in organic functionality. More details about each of these sensors and their combination with the nanomaterials will be the topics of the next classes in the current course. After the sensors of an electronic nose are in place, an algorithm must analyze the responses of the sensors. As a first step, an electronic nose needs to be trained with qualified samples so as to build a database of reference. Then, the instrument can recognize new samples by comparing volatile or non-volatile compounds' fingerprint to those contained in its database. Thus, they can perform qualitative or quantitative analysis. This, however, may also provide a problem, as many odors are made of multiple different molecules. In this case, the device, as it will register them as different compounds, and will result in incorrect or inaccurate results depending on the primary function of the nose. Machine learning primarily concerned with the design and development of algorithms that allow computers to learn and recognize complex patterns based on the data, and this is of course one of the main parts of the electronic nose, as we will detail in the next few slides. The main approaches for machine learning are supervised learning, and it's actually a classification that is seen as supervised learning, and in other words, the data are labeled with predefined classes. In this case, the test data are classified into these classes. The second subcategory of the machine learning is called the unsupervised learning, or clustering. Class labels of the data are known in this approach. However, given a set of data, the task is to establish the existence of classes or clusters in the data. A common unsupervised machine learning that has found applications in the fields like face recognition and image comparison is called Principal Component Analysis, or briefly PCA. It is a common method for finding patterns in data of high dimensions. The aim is to achieve lower dimensional representation and visualization of the collected data in terms of scores on uncorrelated principal components, so-called score plot. This method enables us to interpret a large set of data by using a smaller number of components. Since patterns in data can be hard to find in high dimensional data, and there is no ability to represent this data graphically, PCA is a powerful tool for data analysis. A simple way to understand PCA is how to take a picture of a teapot to capture the most information about it. You can take a picture in several directions, but how you will know which direction contains the most information? Also, what about the information that is lost? Well, the front side of the teapot contains the most visual information. The way to find this visual information is by rotating the pot around its center till the longest axis is revealed. The next step is to find the second longest axis that is orthogonal to the first one. The two axes found are the first and the second principal component in this case. The teapot that had many dimensions now have only two main dimensions that contain most of their information. Supervised learning, or the so-called machine learning, takes a known set of input data and known responses to the data and seeks to build predictor model that generates reasonable predictions for the response to new data. In order to solve a given problem of supervised learning, one has to perform the following steps. The first step is to determine the type of training examples. Before doing anything else, the user should decide what kind of data is to be used as a training set. In the case of handwritten analysis, for example, this might be a single handwritten character, an entire handwritten word, or an entire line of handwriting. The second step is to gather a training set. The training set needs to be representative of the real-world use of the function. Thus, a set of input objects is gathered and corresponding outputs are also gathered either from human experts or from measurements. The third step is to determine the input feature representation of the learned function. The accuracy of the learned function depends strongly on how the input object is represented. Typically, the input object is transformed into a feature vector which contains a number of features that are descriptive of the object. The number of the features shouldn't be too large because of the curse of dimensionality. But of course, it should contain enough information to accurately predict the output. The fourth step is to determine the structure of the learned functions and the corresponding learning algorithm. Now, we will move to the fifth step. And indeed, the fifth step is to complete the design, namely to run the learning algorithm on the gathered training set. In this case, some supervised learning algorithms require the user to determine certain control parameters. These parameters may be adjusted by optimizing performance on a subset, which we call usually validation set, of the training set, or throughout the so-called cross-validation process. The last step in this approach is to evaluate the accuracy of the learned function. After parameter adjustment and learning, the performance of the resulting function should be measured on a test set that is separate from the training set. A wide range of supervised learning algorithms is available, each with its pros and cons. There is no single learning algorithm that works best on all supervised learning problems. Therefore, one has to tailor the algorithms according to the targeted application of interest. In conclusion, the electronic nose system, similar to the human nose, contains a hardware and a software parts. This field is under extensive research and provide opportunities in the fields like food industry, medical industry, and environmental monitoring. We come now to the end of class number 3, session number 4. Thank you.